So I'm going to move from, seems loud, from genetic connectivity to more trophic connectivity and talk about, synthesise some studies that we've been carrying out over the last number of years. So I'm Glenn Hyndes from ECU, and there's a whole bunch of co-authors who are very much integral to this, this study. So back in 2014 we published a paper that looked at trophic connectivity across a whole range of different types of ecosystems. One of those was what we called the sort of coastal, exposed coastal ecosystems, where we looked at areas where there were rocky reefs with kelp and seagrass meadows going into sandy beach areas. And in some of these regions, large amounts of material gets washed up onto the beaches. That material really comprises mainly Clonia and seagrass. So there's a lot of connectivity in terms of the movement of material from the reefs into seagrass meadows and the reefs and seagrass meadows into other ecosystems. We did an analysis of the published data that was out there and showed that both systems are very productive. There's a large level of export from both systems as well. So if we look at reefs, there's around about a third of the material is actually exported out of the reef system. In seagrass meadows, it's around about half of the material is moved out of the system. So what you have here is seagrass meadows that are highly productive. They live generally in these areas in quite oligotrophic waters, so very nutrient poor waters. They're losing a lot of material, yet they're highly productive. So there has to be some mechanism by which these systems are able to sustain themselves. So what we wanted to do was really look at, explore the mechanisms by which they could sustain themselves. Some nice work done by de Bertinis back in 2013 showed that a large amount of material was actually exported from the reefs around southwestern Australia, and a large part of that was through erosion, though I'm not actually going to be talking about that today. This is the bit that I really want to talk about. So large amounts of material is actually dislodged from the reefs and exported into other systems. And Wernberg et al. Back in 2006 showed a large amount of that material can actually lodge itself in seagrass meadows themselves. So that's what I really wanted to focus on. So we're going to be focusing on an area that is oligotrophic, so nutrient levels are, in terms of the bioavailable nutrients, are very, very low, highly productive seagrass meadows. And so we wanted to really explore how the nutrient cycling might take place. So the aim really was to determine pathways of the kelp subsidies in seagrass meadows through the micro level, which is dissolved organic nutrients, and then the macro level and the micro level through the particulate organic material. So these are the potential pathways that we explored, and when we're looking at kelp specifically here, and our study in 2012 showed that there wasn't much in the way of inorganics, nitrogen, that was actually being released from kelp, but there's a large amount of dissolved organic nitrogen that's being released. So we wanted to really focus on, well, what are the mechanisms, potential mechanisms for this material making its way into the seagrass meadows and taken up by the seagrass meadow ecosystem. And part of the DON is urea, which is a small molecular weight DON, which I might come back and talk about a little bit later. So what we wanted to do was really, first of all, look at the dissolved organic nitrogen component and work out whether or not that's actually taken up by seagrass and epiphytes and how that might actually be taken up. What are the mechanisms associated with that? Then look at it more at a macroscopic level and look at the effects of the kelp on the actual mesograzers in the ecosystem. So back in 2012 we published a paper where we looked at, we enriched kelp with ammonium chloride, high level of 15N. We shoved it out into seagrass meadows. We had controls and treatments, and then came back 2 weeks and 4 weeks later to look at the uptake of that delta 15N, or the 15N rather, in seagrass and the epiphytes. What we showed in the treatments was that there was a large amount of uptake of 15N in the treatments after 2 weeks, and then also after 4 weeks as well. So this showed that there was a large amount of nitrogen being taken up by the seagrass and the epiphytes. Following on from that, we managed to co-opt a student, a PhD student, into looking at the microbial community in seagrass meadows. Not a lot has been done on microbial communities in seagrass meadows. So she looked at the actual amount of bacteria and archaea found on the leaves of seagrasses. So here we have the microbes on biofilm, sediment, surface water and water column. So we're looking at different compartments within and above the seagrass meadows. What she showed that the bacteria through 16S RNA were very, very high and much, much larger in abundance than the sediment and also much larger than the surface and the canopy water. So large amounts of bacteria, and similar for the archaea, which we often consider to be a major component of much more extreme environments. And those bacteria and archaea that are associated with the nitrification process were also very, very high. And indeed what she showed was that the abundances of these bacteria, or the microbes in general, were higher than often what is found on kelp and other macroalgae. So very high abundances of microbes, and certainly high abundance of microbes that are able to be involved in the nitrification process. So through a process of nanoscale secondary ion mass spectrometry, otherwise known as NanoSIMS, much easier to say, this shows through imagery the uptake of the 15N. So here we can see after 2 hours in seagrass which has bacteria or microbes associated with the leaves versus leaves that had actually been swiped with antibiotics and scraped of any of the biofilm. We show here that after 2 hours there's some uptake of the 15N into the cell wall, and then we're seeing it going into the cytoplasm, the chloroplasts of the actual cells themselves. So we can see the uptake of 15N by the seagrass cells. Whereas where there were no bacteria or no microbes, there was no uptake whatsoever. So clearly the microbes are playing a role in allowing seagrass to take up nitrogen. In this case, seagrasses aren't very good at taking up dissolved organic nitrogen, but they are much better at taking up dissolved inorganic nitrogen. And through that experiment, previous experiment, and another experiment, what was concluded was that the microbes break down the dissolved organic nitrogen into ammonia and then also into nitrate, and that nitrate and ammonia are then taken up by the seagrass, go into the actual cells of the seagrass, and then make their way into the cytoplasm, and then eventually they can produce their own amino acids. So there's a clear mechanism here for how seagrass can actually take up nitrogen, dissolved organic nitrogen, and clearly microbes are a part of that process. So that's the micro scale. Now we're going to be looking at the macro scale, and hopefully I've got a little bit of time to do that. Again, in 2012, we showed through putting out enriched kelp that the mesograzers were able to take up nitrogen, the 15N, from this enriched eclonia. Later on, we decided we would then do some growth experiments for these mesograzers, feed them on kelp, feed them on epiphytes, and then feed them on a combination of both. What we showed was that when they were fed on kelp, there was actually very little growth associated with that as a food source. Because they were fed on epiphytes or a combination of epiphytes and kelp, then growth was much, much greater. So there was a significantly greater amount of growth when they were fed on epiphytes alone. But if you looked at it from an abundance perspective, then when you put out kelp, then there was actually much, much larger numbers of these mesograzers. In this case, the cantharides, which is the mesograzer, much, much higher in those areas where kelp is present at a range of different sites compared to those areas where kelp was absent. And the same for this Palomnetes species here as well, much higher abundances. And interestingly, these numbers here, 68, 86, 71, actually indicate the number or the percentage of animals that were actually found on the kelp itself. So clearly they're attaching themselves to the kelp, but that growth experiment suggests that they're not actually gaining anything from the material itself, but what they could be doing is gaining protection from predation rather than necessarily a food source. So, in conclusion, we looked at the micro and the macro scale here. In terms of the micro scale, then dissolved organic nitrogen, which is leached out from kelp, is then processed by the microbes, by both bacteria and archaea, producing DIN in the form of both ammonium and nitrates, which can then be taken up by seagrass and potentially increase production of seagrass. And I guess the production of the seagrass, the enhancement of the production of seagrass will depend partly on the amount of kelp that is actually making its way into the seagrass meadows and then remaining in that system for a while. At the macro scale, then the kelp can provide a habitat. It's often quite hard to disentangle between habitat and food availability, but what we're suggesting is that the food itself, kelp as a food itself, is probably not that important. It's probably the epiphytes themselves, and what we've shown is that the epiphytes are taking up nitrogen from the kelp, and so there's a potential enhancement of the amount of biomass of epiphytes that's available from kelp, which is then providing a greater food source for the mesograzers. But the kelp itself can also provide a habitat for fish and for the mesograzers, potentially protecting those mesograzers from higher levels of predation. So there are a number of mechanisms by which that kelp might then actually facilitate an increase in the productivity of a seagrass meadow, both by seagrass itself and also its inhabitants. And this work couldn't have been done with the help of a whole range of different people. And so I acknowledge those people, thank them very much for their help with the work. It's been a whole range of different work coming into this, and of course they're not the only people who've been assisting with the work. There's been a whole bunch of other volunteers and supporters, so thank you very much, and thank you for being here. Questions? Yeah, Gary. Both. They can be feeding on epiphytes, as in the macroalgae, and they'll be also feeding on the more of the periphyton material as well. So when I was talking about epiphytes there, I was talking about the actual macroalgae rather than the periphyton, but they will be growing on that as well, feeding on that, yeah. And that will also be taking up, in fact, the NanoSIMS results show that there's a large amount of uptake of 15N associated with the DON from kelp by that periphyton as well.
How does nitrogen, which gets very little attention in a general sense, nitrogen fixation, this association either with on the leaves or the symbiotes within the root rhizome system, play in bringing up the addition of nitrogen to seagrass leaves?
So nitrogen fixation is certainly part of it. We didn't look at that because there has actually been more focus on the nitrogen fixation. And we felt that there was another player in that field as well. And now that's really probably the next step, because what we're showing is that there's a large amount of processing actually around the leaves of the seagrass. That nitrogen fixation is more likely to be taking place, a large part of it in the sediments. Yeah, that's right. So I think the next step would be to try and get that nitrogen budget associated with that fixation as well. Yeah. Good point.
Cowan, are you prepared to go as far as to say that the kelps are contributing to blue carbon?
God, blue carbon! Are they contributing to blue carbon? Well, yes, of course they will. The proportion at which they're doing that is the next question, I guess.
I think your evidence would support, but it needs to be further done.
Yeah, and I think it's an indirect effect. So, I mean, kelp, when we're talking about blue carbon, we're thinking about those plants that are actually sort of got root systems and binding things up. But I think that here we're showing that that material coming in, decomposing, will actually contribute to productivity, and then you're contributing to blue carbon. So yes.